of the digestive tract. All right? So let's talk about functions of saliva. First of all, saliva forms a protective covering in the oral cavity. So it covers, it sort of coats the oral mucosa. It also buffers. You're probably aware, and if you're not, you will be, that bacteria tend to create an acidic environment in the oral cavity. So saliva is made up primarily of mucus. There's some serous secretion as well, but it tends to have a more alkaline pH to neutralize some of those acids that you find in the oral cavity. And then each tooth is covered by a thin layer of protein, which we call a pellicle. So that is, the saliva helps to form the pellicle, covering the tooth, and also helps to maintain the integrity of the tooth. And saliva has antimicrobial properties, certain chemicals in the saliva, and aids in hastening tissue repair if you have any lesions in the oral cavity. And then digestion begins in the oral cavity with saliva. So saliva does several things. One is to moisten the food so that it's easier to break it down into smaller particles, but it also contains enzymes such as salivary amylase that can begin breaking down carbohydrates and so forth. So digestion actually begins in the oral cavity. And then the sense of taste is dependent on saliva. The things that we taste need to sort of dissolve in the saliva to make them more accessible to our taste buds. So saliva has a lot of important functions. You will have courses, if you haven't already had so, where you go into detail about the properties of saliva, the components, and so forth. And you'll talk a lot about xerostomia, which is the side effect of many medications and diseases where your mouth doesn't produce enough saliva. And you're much more prone to caries and other problems in the oral cavity if you don't have enough saliva. Okay, so there are 3 pairs of major salivary glands. We say major because these are glands that you can see macroscopically. They're large. And so the largest of the 3 is called the parotid gland. Otid refers to the ear, and parotid means next to the ear, so that's where the name comes from. If you look at this picture from Netter's Atlas, you'll see the ear canal. Now the outer ear has been removed, but here's the external auditory meatus, the canal, and you can see the parotid gland is located just anterior to that. So this is the parotid gland. It's the largest of the major salivary glands. And the facial nerve, which we haven't studied yet, but it's the nerve that innervates the muscles of facial expression, passes right through that gland. So if a physician needs to do a parotidectomy, where they have to remove the parotid gland, what they have to do is remove the parotid gland piecemeal, being careful not to damage the branches of the facial nerve that are passing through it, because if you damage that nerve, then you're going to get facial paralysis. These muscles will not work anymore, and you'll droop. Okay? The duct of the parotid gland opens in the oral cavity opposite the second maxillary molar. So if you look inside your cheek, you'll see this little bump, and that's where the duct is opening up. The parotid gland is innervated by cranial nerve 9, the glossopharyngeal nerve, and that leads preganglionic parasympathetic fibers to the otic ganglion, which is in the infratemporal fossa. You don't need to know this now. We'll go into detail next quarter. And then coming from the otic ganglion, there's a nerve called the auriculotemporal nerve that brings postganglionics to the parotid gland to stimulate secretion. The second group of salivary glands, one on either side, is called the submandibular gland. In a normal healthy person, even though they're located right here, you can't really palpate them. You palpate them when they become inflamed, enlarged, or if they have stones or something in them. Otherwise, they're very soft and they don't palpate very well. So there's 2 submandibular glands. They're located in the posterior part of the floor of the mouth, and the duct opens under the tongue. You know these 2 little bumps where you can bleep? Those are the openings of the submandibular duct. We call those sublingual caruncles. So the caruncle is that little bump with a hole in the middle where saliva squirts out. Some people can squirt saliva on command. That's one of my fantasies. I can't do it. I wouldn't make it. I am jealous of people that can do that. I do it accidentally when I go to the dentist. I think that's where they wear all their body armor. You know, they have masks and gloves, and I don't know. I feel like I must be contaminated because they've got all this protective body wear on, but it's probably so I won't squirt on them. Anyway, the innervation of this and the next gland come from the facial nerve, which is cranial nerve 7, and then that a branch of the facial nerve joins with the lingual nerve, which supplies sensation to the tongue, and then from there you get preganglionic parasympathetics going to a ganglion called the submandibular ganglion, which in this picture is right there, and then postganglionics from that ganglion are going to go to both the submandibular gland and the next gland we're going to talk about, which is the sublingual gland. So here are the sublingual glands, and notice this is another picture from Netter. This shows all 3 of the glands. So here's parotid, here's submandibular. Sublingual is right under the tongue, which is why it's called sublingual. It's the smallest of the 3 major salivary glands. It's located in the floor of the mouth, lateral to the tongue. It has lots of little ducts that open up, and it also secretes into the submandibular duct. And its innervation is the same as the submandibular gland. Now, Dr. Banks will be lecturing on the head and neck stuff, and so he'll give a lecture that will include the salivary glands, and he'll repeat all this information that I just told you. So I'm just giving this to you to sort of help you get into the salivary groove here. Now, there are also minor salivary glands, and these are little glands that are embedded in the mucosa of the oral cavity. So they're not major structures that you can see grossly. You can see them histologically under a microscope, but grossly you don't see them. So they're in the mucosa, lining the cheek, under the tongue, the roof of the mouth, there's a lot of them, and so forth. Okay, so there's between 600 and 1,000, I don't know who counted all these things, but this is an estimate, of the minor salivary glands. They're very tiny. They're present throughout the oral mucosa. The only place you don't really see them is the gingiva, covering the bone, where your teeth are, and the anterior part of the palate. So in this picture from Netter's atlas, Netter has peeled away the mucosa, but he's left the glands on one half of the palate so you can see them. So notice it's just packed with these little glands. They're mostly mucus. There's some serous acini as well, but there's more mucus than serous. The one exception is a gland on the tongue, or a group of glands on the posterior part of the tongue that are called von Ebner's glands. Now remember, I always tell you we're trying to get rid of people's names, but we haven't gotten rid of von Ebner yet, whoever he is. So associated with some, we have some papillae, which are called circumvallate papillae. You'll learn about those later. Surrounding those papillae and draining to the moat around them are these serous glands, named the glands of von Ebner. This shows up on exams quite a bit, so you want to remember that the glands of von Ebner in the posterior part of the tongue are pure serous glands, whereas most of the other minor salivary glands are mucus. How do salivary glands develop? Well, as the oral cavity is developing, the oral cavity is lined by epithelium, which derives from ectoderm. So we call it oral ectoderm or oral epithelium. And as the epithelium grows, there are little outgrowths or outpocketings of this epithelium that begin to grow down into the mesenchyme surrounding the epithelium. And this is the beginning of the salivary glands. Now, salivary glands, since they are exocrine, have ducts, as we've already seen, and so those ducts are going to maintain their contact to the epithelium from which the gland grew. The mesenchymal cells surrounding the little glands will condense around the gland and form a capsule around the gland. The type of growth that we observe in salivary glands has been called branching morphogenesis. Why do we call it that? Morphogenesis, of course, means the development of shape or form, the development of the gland. And so as the gland grows, it undergoes multiple branching patterns. So dichotomous branching. So it will grow down, that will branch, then those branches will branch, those branches will branch. And so that's what we mean by branching morphogenesis. All right? Now, when salivary glands first start to grow down into the mesenchyme from the epithelium, they are solid cords of cells. There's no lumen. And so once the salivary gland has started to develop, lumens begin to open up. But you would think that the lumen would open up proximally and then go distally, but it's just the opposite. The first lumens to appear are those that are farthest away from the surface epithelium, and then it goes backwards, and then that part that connects to the epithelium opens up last. So as the lumen opens up, surrounding the lumen there will be two layers of cells. The inner layer is going to differentiate into secretory cells, and the outer layer is going to differentiate into myoepithelial cells and into the connective tissue capsule and into the various septa. So here is a picture of a developing salivary gland. MES stands for mesenchyme, and so all of this tissue is mesenchymal tissue around here. And then you can see the various branch points as this structure branches, and you can see that parts of this have already gotten excavated and have a lumen in them, but parts of them the lumen hasn't opened up yet. Now in this particular picture, I don't know exactly what the orientation of this picture is, but we don't see the surface epithelium from which this gland developed in this particular picture. It's out of the plane of section, but you can see the developing stuff. And so here is a closer view, and here you can see that there are two layers of cells. So here's the inner cell layer, so those are going to become secretory cells, and then here's the outer cell layer, which is going to be support tissue. And here you can see the lumen has already formed, but down here there's not a lumen yet. Here you can see several different branch points. So the arrows are pointing to some different branch points. So you can see the branching morphogenesis occurring. And then this tissue out here is mesenchyme. Right. Okay, so that's just a little bit about how salivary glands develop. Now let's talk about the actual histology of the salivary glands. Salivary glands are acinar type glands. During the first part of the quarter, we looked at the difference between serous and mucous glands, and we learned about, sebaceous glands and sweat glands. So forget sebaceous and sweat. Those are out of the picture. The only kinds of glands we will see in the oral cavity are serous and mucous glands. All right? We won't see other types of glands. And so both the major and the minor salivary glands are made up of serous and or mucous acini. Right? Remember, acini are little balls of cells that have a lumen in the middle, and then coming from that lumen, you're going to have a duct. Now, in the pancreas, as we saw yesterday, there's centroacinar cells inside of the lumen, which are the beginning of the intercalated duct. We don't see those in the salivary gland. So here we won't see the centroacinar cells, but we still see a lumen, and the lumen still opens into a duct. All right? So the part that secretes is called the secretory end piece. That's sort of a synonym for the acinus. And so here is a picture from a textbook showing various types of secretory end pieces. Here we have a serous one where all the cells are serous. This one is mostly mucus here. So, whoops, I always do that. I hit the button accidentally. That's why I can't use the mouse. Okay. So this one is a mucus one. Notice the mucus acini tend to be more tubular in shape, whereas the serous acini tend to be more spherical in shape. So there are slight differences in shape. And then surrounding the acini, we have these myoepithelial cells. We learned about them previously. They can contract, so forth. And then the acini are all going to drain to ducts, which drain to larger ducts until the saliva is secreted. By the way, the word acinus comes from a Latin word which means a barrier or a break or something like that. All right, so here's a nice little picture showing each little orange dot is an acinus, and then the little yellow things, of course, are the ducts. And you can see there's smaller ducts that lead to larger ducts and lead to larger ducts. And then each gland is surrounded by a capsule. Extending into the gland from the capsule are septa, and the septa contain blood vessels and nerves and so forth to supply stuff for the formation of saliva. Here is a nice drawing of an individual acinus. So these orange cells are the secretory cells, and the yellow cells are the duct cells. And you can see that the nuclei are basally located, spherical, euchromatic. The apexes of the cells are filled with secretory granules. The contents of those granules are going to be released by merocrine secretion into the lumen. Remember we learned about 3 different types, holocrine, apocrine, and merocrine. So salivary glands are all merocrine. All right. So, we said there are 2 different types. There's serous and there's mucus. And so the serous cells produce a fluid that's rich in proteins, glycoproteins. The acini tend to be spherical in shape. The cells, because they are in the wall of a ball, the cells tend to be broader at their base and narrow at the apex, so they're sort of pyramid shaped. The nuclei are spherically located and spherically shaped and located at the base of the cell. Secretory granules fill the apex and contain macromolecules of various types, whatever the secretion product is. Lots of rough ER, Golgi, mitochondria, lysosomes, and all that kind of stuff. All right. Now, typically, serous cells have a few small microvilli on the surface. They interdigitate laterally. What that means is that where one serous cell is in contact with another serous cell, the membranes are not straight, but they go like this. And so that increases the surface area, increases the attachment between the cells. I can see people going to sleep, so let's take a 30-second break and just stand up and stretch. One person, 2. Oh,
stand up. That's right.
I think it increases surface area.
Hey, I'm gonna need you to stand up, young man.
Yes, you can.
Look at that.
okay.
Nina, just because you're 21.
Sit down.
Oh, you came back?
Yeah, I have a very short attention span, so if I wasn't talking, I'd probably be sleeping as well. I put myself. All right. In addition to the lateral foldings, we also have basal foldings. And so obviously the basal foldings are going to increase surface area. Whenever you have a cell where the basal membrane is highly folded, typically that indicates that there's going to be fluid transport. You want a large surface area so you can transport the maximum amount of fluid. Saliva is water-based. You need that fluid to come from somewhere. And so you'll typically have lots of pumps in the basal membrane, and you'll also transport large quantities of fluid. So that's what the basal folding has to do with. There are junctional complexes. There are hemidesmosomes attaching the basal part of the cell to the basal lamina. There are gap junctions between the cells, which helps to coordinate their function. They communicate with each other. And these cells have intercellular canaliculi. Now be careful, because when we talked about parietal cells the other day, they have intracellular canaliculi. Rather than being intracellular, what this means is that between adjacent cells, you have this extension of the lumen down in between the cells, and we call that an intercellular canaliculus. Okay? So don't confuse the two, they're different. And this obviously is going to increase the surface area for the release of saliva from these cells. All right, so here's a diagram from a textbook showing the basal folding, the lateral interdigitation, junctional complexes, and then here is an intercellular canaliculus. So you can see that it extends most of the length of the lateral domain and allows this cell to have more surface area to release its product than it otherwise would. And then most of the organelles are located in the basal part of the cell, because the apex of the cell is filled with secretion granules. And then here's an electron micrograph showing it. So you can see again the secretion granules here. You can see this large nucleus at the base, lots of rough endoplasmic reticulum, and here is an intercellular canaliculus right here. And the lumen is up here. All right, here is a low power micrograph showing a sagittal cut through an entire salivary gland. I don't know which gland this happens to be. I'm going to guess that this is the submandibular gland, because I see lots of mucus and serous acini. If this were the parotid gland, we would see almost completely serous. We would hardly see any mucus, if any at all. But the submandibular gland is a combination of mucus and serous, and the sublingual gland is mostly mucus. So each one has a little bit of a different composition to it. I'm going to guess that this is submandibular. It doesn't really matter what it is. The point of this slide is to show you that there are lots of these connective tissue septa, dividing the gland up into little lobules. And of course, what isn't shown here is that in these connective tissue septa, there are blood vessels, nerves, lymphatics that are supplying the gland. We don't want to just supply the outside of the gland, we want to supply the inside of the gland as well. So that's the function of these septa. There are larger ones and smaller ones, large vessels and smaller vessels, large ducts and small ducts, and so forth. And then the acini are these little guys out here. The light-colored things are going to be mucus. The dark-colored ones are going to be serous. All right. So the mucus cells then produce mucins, and these are rich in glycoproteins. Now, this little guy is pathology. Mucus is a normal, healthy production of our glands. But this kind of mucus, which is produced when you're sick, is pathological mucus that has bacteria and stuff in it, so it's different from normal mucus. So we're going to ignore him and just look at the normal healthy mucus. And this lubricates the oral cavity, forms a barrier that helps to prevent infection, binds and aggregates various microorganisms like this guy right here, and the secretory end pieces tend to be more tubular in shape rather than spherical. Mucus acini are often associated with serous acini in the form of serous demilune, so that's what you're seeing here. These are the mucus cells, these out here are the serous cells. And demilune refers to the shape of the serous component. All right, the apical part of the cells contains large quantities of mucin, big vesicles. The nucleus and the refiar are compressed down in the basal part of the cell. In a routine H&E section, the cells appear to be empty because mucus doesn't stain. And so you need special stains like that previous slide to show the mucus, otherwise it just looks white, as you've already seen. Cells are joined by junctional complexes, but mucus cells do not have intercellular canaliculi, so it's only the serous cells that have that, not mucus. Here's a cartoon from the same textbook showing the mucus acinus. So these are Golgi apparatus, the blue. The green is the rough endoplasmic reticulum. Notice the nucleus has a bit of a different shape, and lots of big granules containing mucins that are going to be released out of the apical part of the cell. And here's what it looks like with an electron microscope. So you can see these large aggregates of mucus granules, whatever they're called. Nucleus way down here at the bottom, and the lumen is up here. And then this is what it looks like at the light microscope level. You're familiar with this. You've seen it. You've got to be able to distinguish mucus from serous, from sebaceous, from sweat. So make sure you can differentiate between the different types of glands. If you can't, get some slides, go online, look in textbooks, figure it out. And if you need help, I would be glad to help you. All right, myoepithelial cells, we've already mentioned those previously. They are contractile cells that have large extensions, sort of like tentacles, that surround the acini, and they can contract and help to move the fluid along. They are derived from epithelium. They're not derived from mesenchyme. So that's why we call them myoepithelial cells. They surround the acini, they surround intercalated ducts, and they're located between the basal lamina and the secretory cell. So they are epithelial in origin, which means they're inside of the basal lamina. They're part of the epithelium. So the basal lamina is external to the myoepithelial cells and to the secretory cells. So they're just in contact with the secretory cells. They're not separated from them by a basal lamina. They attach by hemidesmosomes. They have multiple processes and so forth. What are their roles? We're not sure. We know that they provide support for the secretory cells. They help to expel saliva. They help to maintain patency. That is, they keep the ducts open. That's what patent means. And they also provide signaling molecules of some sort to the acinar cells. We're not sure exactly what. It's thought that they may have tumor suppressor properties. They have proteinase inhibitors. They have anti-angiogenesis factors. So here's two different separate pictures. One showing an entire secretory end piece with myoepithelial cells surrounding it, and another showing the fact that here are the secretory cells. They form hemidesmosomes, or desmosomes rather, with the myoepithelial cells, and the basal lamina is outside of the myoepithelial cell. And then here's another picture. This is an immunostain that has been, so we raised antibodies against a specific protein in the myoepithelial cell to demonstrate it. And then here it is in electron microscopy. So this is the myoepithelial cell here. These are the secretory cells here. And here's another picture. Boy, I'm really beating you over the head with myoepithelial cells. So this is a scanning electron unit. Okay, let's talk about the ducts. Not that kind. All right. We have the first smallest ducts are called intercalated ducts, just like we saw with the pancreas, because they're located between the acini. And so they begin within the acinus. They receive what we call primary saliva. So the saliva is going to be modified as it flows through the duct. What comes out into the oral cavity is not the same composition as what the secretory cells produce. So as it passes through the duct cells actually modify the content of the saliva. So what the secretory cells produce is called primary saliva. It's isotonic with blood. It will not be isotonic by the time it ends up in the oral cavity. So intercalated ducts are lined by simple cuboidal epithelium, the central nuclei, and blah, 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 blah. You can read that, and they have myoepithelial cells, and they add lysozyme and lactoferrin to the saliva. So they do have a secretory role. Okay, and once again, these are the intercalated ducts, these very first ducts that come from the acini. And then here is a duct cell. You won't be tested over this. This is what a duct cell supposedly looks like. Intercalated ducts are going to lead to larger ducts, which we call striated ducts. The reason why we call them that is because when people were first studying them with the light microscope, it appeared as though the basal part of the cell had striations in it. The reason for that appearance is that the basal part has these big folds, and between the folds there are rows of mitochondria, and so at the light level, that gives a sense of being striated. They're not truly striated like striated muscle, but they have that appearance, and so that's where the name striated duct comes from. So these receive the primary saliva from the intercalated duct, but it's in the striated duct where the composition of the saliva is modified. So these are the cells that modify the composition of the saliva. They form the major part of the duct system. The cells are more columnar in shape, and this is their, you can read through this. So the striations equal the mitochondria lined up between the basal foldings, and they're surrounded by basal lamina, and they're also surrounded by capillaries. Why are they surrounded by capillaries? Well, obviously the cells need a blood supply, but also, since you are changing the composition of the saliva, you've got to be able to exchange that stuff with blood. So you're taking some things out of the saliva, putting them into the bloodstream, taking things out of the bloodstream, putting them into the saliva to change the composition. So whenever you do that, you need to have capillaries surrounding the ducts. And so they contain secretory granules, they have lysosomes, peroxisomes, tight junctions, and all kinds of stuff, but no gap junctions. So here are some acini, here are the intercalated ducts, and then here are the striated ducts. So you can see that the base of the duct looks like it has striations. And that's where the name striated duct comes from. And then here is an artist's conception of the basal folding with the mitochondria lined up between the folds. So that gives the striated appearance to the cells. And then the striated ducts lead to the bigger ducts, which are the excretory ducts, the ones that actually release the saliva into the oral cavity. And these are located in connective tissue septa between the lobules. Their cells are pseudostratified or stratified, and so forth. They have special cells called tough cells, which have very long, stiff microvilli. And we won't ask you to identify the various cell types on the practical, so you don't need to worry about that. Here's a diagram that just shows the various levels of ducts, the various sizes and locations of the various ducts. Okay? Then we have the capsule surrounding the whole gland. The capsule is connective tissue, collagen, type 1 collagen, fibroblasts, typical connective tissue. And then extending in from the capsule are septa, which divide the gland into lobules and transmit blood vessels and so forth. And so these are the cells that are found. Hello. I'll be done in just a minute. Come on in. These are the cells that are found in the septa and collagen and elastin fibers. Okay, we will stop here. You have a 10-minute break, and then you'll have Dr. Escobar talking about posterior abdominal wall. Is that right? Posterior abdominal wall. Woo-hoo. All right. Have a wonderful weekend. See you next week. And so my quiz questions on Tuesday will stop right here. I won't ask anything else.
Wait, not even a
And we'll
double.
finish this lecture up during lab time.
Oh,
What?